All right, welcome everyone. I apologize for a bit of a late start. I was told the parking lot was filled to the brim, so we were giving latecomers a little longer to get here, but well, we're off and running now. So I'm Sandy Williams, president of the Gladstone Institutes, and I offer each of you a very warm welcome. I'd refer you to your program because therein you'll see a number of people very important to Gladstone listed there. Our Board of Trustees, our Gladstone Foundation Board, and our President's Council, and many of these individuals are with us tonight. The other message, though, is everyone here is important to us, and I'm grateful that you're here. One of the, I think, privileges I'd like to think of being in the Gladstone family is every now and then we like to offer to you a premier seat for important things that are happening right now in science. And I'd like for you to think about the experience that we'll all enjoy for the next hour or so in that light. I think most of you are aware that over the last 10 years or so, accelerating over that time, there's been what can quite rightfully be called a revolution in science. That's not an exaggeration, and I think you understand that. This revolution goes by the name of stem cells or stem cell biology, and we've chosen as the title for this experience today, From Stem Cells to Cures. I want us to be sure, though, to frame this up, that we have a clear understanding of what we mean by those words. I think when referring to stem cells, there's lots of different kinds of stem cells. There are embryonic stem cells that in humans can only be derived from human embryos. There are other kinds of specialized stem cells like cord blood stem cells or bone marrow stem cells. These cells arise naturally, and we've known about such cells for a while. What's been revolutionary more recently is the ability to do what we call cellular reprogramming, going into cells and rewriting the code that nature uses to specify why one cell is like this and another cell is like that. And this cellular reprogramming is what has really launched the stem cell revolution. We're quite pleased and gratified that a good part of that revolution has taken place right here at Gladstone. And without question, the pioneer and leader of that has been Shinya Yamanaka, who will be one of our speakers in the conversation tonight. So there is a revolution. It concerns stem cells. To go a little bit further, so we're all on the same page before we start, I'd like you to be aware that there are at least 3 pathways by which advances in stem cell biology or cellular reprogramming can be turned into new medicines or other ways that would affect human health. The first is the most obvious way, and I think this is what people tend to think about when they think about medical uses of stem cells. You take cells out of the body and then you put them back in. You transplant cells into the body. This is standard medical practice now for bone marrow stem cells, but other kind of applications are beginning to emerge. Some of these are going to be promising. Some of them are, quite frankly, bad medicine. There was an article in the New England Journal of Medicine this week about stem cell clinics popping up all over the country and the world, where much of what's passing as stem cell medicine is not legitimate. And it is not likely to lead to medical advances, and at present it's unregulated. That's not what we're talking about tonight. We're talking about the true important advances that could come from the use of stem cell knowledge and cellular reprogramming to create cures. But nevertheless, the notion of taking cell out of the body, doing something to it, in this case reprogramming it and putting it back in, is real here and now. There are clinical trials of that kind of approach being conducted. We gave an award here a few weeks ago to a pioneer in that field from Japan, and I'm sure our speakers tonight will address this. But please be aware that there are at least two other paths by which advances in stem cell biology can promote cures. One approach is not to take cells out of the body, reprogram them, and put them back in for some good purpose. The other approach is to send signals to cells that are already in the body to change into something they otherwise wouldn't do. This is a separate path to restoring the function of damaged organs, the heart, the liver, even the brain. So please be aware there's another approach, and one of the pioneers of that approach is also with us tonight, Deepak Srivastava, and I'll call on him later in the evening. There's a third approach, though, that in many ways may be the fastest way that stem cell biology leads to cures, and that's taking cells out of the body, skin cells, for example, turning them into a different kind of cell outside of the body in a dish. Many of you have been upstairs and seen the beating heart cells that are made out of skin cells, which we do routinely now. Those kind of cells outside of the body can become, not yet there, but we believe can become very important and powerful models to find new medicines and validate new medicines. So cellular transplantation, reprogramming of cells inside the body, and cellular models of human disease created by cellular reprogramming are all different routes to cures. Now, to conclude with my preamble, I've defined for you what we mean by stem cells, and then I want to move to the cure end. 2 points there. The best science will never be a cure unless it passes into the commercial arena. It has to pass through venture investors, biotechnology companies, pharmaceutical companies, device companies for scientific advances to help human beings. So necessarily, nonprofit scientists like Gladstone must always find our partners from the commercial world, venture, biotech, pharma, and so also tonight we have Blake Byers, who represents that sector of what must be teamwork to move from stem cells to cures. And then finally, let me define what I mean by cures. I used to be reticent to use that word when I talked about what my lab does or what my colleagues do. I felt a little embarrassed to say we're out curing disease, because in fact, such cures are rare. They are rare. But the beauty of modern medicine is they are happening now. And when I use cure tonight, and I think our speakers will use this broad definition too, I mean any advance in medicine that could prevent a disease that we can't prevent now, could take a chronic disease that is ruining someone's life and make that life better, or actually eradicating the disease from the person's body or even from the face of the earth. When I say cure tonight, I mean all of those things. Fortunately, I think we have medicines that are coming out today that are real cures, all the way to that last ultimate eradication of disease from the earth. But I think we may have to take some more modest steps to get there. So from stem cells to cures, we have a great experience for you tonight, and I'll ask Shinya Yamanaka to lead this off. We won't make long introductions. They're written in your program, but Shinya has earned virtually every accolade and honor that the scientific community has to offer, so we're pleased to have him tonight.
So thank you very much, Sandy, for your introduction. And ladies and gentlemen, thank you so much for joining us today. I am Shinya Yamanaka. So okay. So, I am now a scientist, but I started my career as a doctor, surgeon, 30 years ago. And it was my father who talked me into medicine. This is my father. Without his support and encouragement, I wouldn't be here today. I respect him a lot. I like him a lot. Especially, I like his hair. To be honest, I'm envious. My father was not a doctor. He was a business person. He had a small factory in a small city in Osaka, Japan. So when I was born, he must have been very happy by having his own successor. But when I became a junior high student, he started saying this way, Shinya, you don't have to take over my business. Instead, you should become a doctor and help patients. I don't know why he said that. Maybe he thought his own son was not talented as a business person. His son was not brave enough to be a business person. I don't know. But anyway, I listened to him and I became a doctor. My father was very delighted. I still remember. But soon after I became a doctor, my father passed away when I still needed him a lot. Before he died, he must have been in pain. But he seemed to be happy, even smiling, by receiving some small medical procedures from his own son. He must have been very proud of his son becoming a doctor. However, I myself felt miserable. I felt useless because I was not able to help my own father as a young doctor. I was not help many other patients as well. Then I became more and more interested in medical research because I thought it would be medical research that could help patients suffering from intractable diseases and injuries. Patients like my father, not immediately, but in the future. That's why I decided to become a scientist. I went back to graduate school, and after receiving my PhD, I came to San Francisco to join Gladstone Institute in 1993. I became a postdoc to continue my training as a scientist. I had a clear vision. I wanted to help patients by doing medical research. But of course, it was easy to say, but very difficult to achieve. I spent many hours, many days, and many years in laboratories without significant success. 20 years later, however, I became extremely lucky to have a wonderful group of people. And that group developed a new technology. Our group was able to find a way to make a new type of stem cells, which we designated iPS cells. With this technology, we can convert your blood cells or skin cells into stem cells, iPS cells. The procedure is very simple. All we need is a small amount of your blood cells, just 4 or 5 ml. That's all we need. And all we have to do is also very simple. we just need to introduce a small number of genes into your blood or skin cells. That's it. After a month or two, we can have your own iPS cells. Well, the procedure is simple, but iPS cells are very powerful. We can expand iPS cells as much as we want. We could have millions, we could have billions of your iPS cells. And after expanding cells, we can convert your iPS cells into many types of cells, such as brain cells, heart cells, and liver cells. Another important aspect of this technology is that cells are rejuvenated during the process of iPS cell generation. So you may be in your 50s, 60s, 70s, even more, but your iPS cells are basically 0-year-old, just like babies, because they have been rejuvenated. So that means if we make iPS cells from patients suffering from brain disease, such as Parkinson's disease, and if we make brain cells from iPS cells, those brain cells are not sick. Brain cells in the patients are, of course, sick. However, brain cells from patients' iPS cells are not sick because they have been rejuvenated. They are cells when the patient were babies. So this means by using iPS cell technology, we can prepare a large amount of healthy brain cells from patients suffering from brain diseases. So now, we can use these cells in two major medical applications. First, we can transplant those healthy brain cells back into patient's brain to obtain functional recovery. This approach is known as regenerative medicine or cell therapy. We have been trying to apply this approach, cell therapy, to many diseases and injuries. For example, eye diseases such as macular degeneration, brain diseases such as Parkinson's disease, and also spinal cord injury, heart failure, liver failure, diabetes, many diseases. Also, we have a similar approach. We have been trying to make immune cells, lymphocytes, that attack cancer cells from iPS cells. So this is going to be a new cancer therapy. This is the first medical application of iPS cells. Another, yet equally important application of iPS cells is in drug discovery. Instead of transplanting back into patients, we can use iPS cells and brain cells or heart cells derived from iPS cells in laboratories. Laboratories of universities, laboratories at Gladstone Institute, or laboratories of pharmaceutical companies, in order to make disease models, in order to perform drug screening. By using this approach, we have been working very hard to find a new cure, new medicine for many diseases, such as Alzheimer's diseases, ALS, and many so-called rare diseases or orphan diseases. So thanks to this new technology, I believe I'm one step closer to my vision. We really want to bring iPS cells to patients. We really want to help patients by using iPS cells. Of course, we still have a long, long way to go. We need to overcome many hurdles. Well, my father was very happy when I became a doctor. But soon after he passed away, I escaped from clinics and became a scientist. So I often think that my father may be upset about my career change. He may be saying, "Shinya, stop shaking test tubes. Go back to clinics, help patients." I can hear it saying somewhere. So I really want to, and I really need to bring iPS cells and reprogramming technologies to patients as quickly as possible before I can meet my father again in the future. Thank you very much. So let me introduce Blake. Blake is very talented, not only as a scientist, but also as a business person. So Blake.
So how many people really quickly ever have that reoccurring dream of where you show up to school and you're about to sit down in your chair and then you find out it's actually the final exam and you haven't studied or prepared yet? So yeah, that's kind of how I feel right now, except imagine the teacher is the Nobel laureate in the field. That's your favorite field, and that's a little bit what this is like. So I'm just going to pull up some notes here, but I don't have the same fun visual prompts. No, in all sincerity, thank you all for having me here today, and thank you to Sandy and the Gladstone family for hosting. This building has many remarkable scientists in it, and it's through their kind of tireless effort that the sphere of human knowledge expands slowly each day. And having been someone at the bench, I know it takes about 99 experiments of failure before you have each one of success, and it's a long and tireless process. But it's really how society moves forward. So I appreciate all the work that they do, and it's really an honor to be here. My first introduction to the stem cell biology field, I was more of an engineer by training, but it was really Dr. Yamanaka's work in 2007 when he published his seminal paper in human cells that caught my eye. I was like, oh, that seems like something that's going to be a pretty big deal. Maybe I should try to work on that and for this whole basic engineering thing. But at the same time, there was another researcher in Wisconsin named Thompson who published a similar paper at the exact same time in another journal, but with slightly different factors. So whereas Dr. Yamanaka had 4 factors on a retrovirus, which is a specific backbone of virus that they were using to reprogram the cells, this other researcher was using a slightly different set of factors on a lentivirus. And this other researcher, Jamie Thompson, he had originally discovered and isolated the first human embryonic stem cells in 1998. And so being a young scientist at the time, I was like, well, I've heard of this Jamie Thompson guy. I've never heard of this Shinya guy. I should just copy Jamie Thompson's work. And so for the next 6 months, I attempted to copy Jamie Thompson's work and was fraught by failure for 6 months, which I just thought was part of the process. Until finally one day I tried Dr. Yamanaka's method, and it worked the first time I tried it. And so I learned my lesson, and now I'm just going to copy Dr. Yamanaka's work from now on. And so then we were off to do the actual science. We had just replicated the work, and we started using the technique to research a variety of different diseases. So we started in a few early childhood developmental diseases and eventually started working on Parkinson's disease as well. And we published a few papers in that field looking at taking cells from genetically defined Parkinson patients. So this is a small subset of patients that have a genetic mutation that converges some type of risk for developing the disease. And we weren't exactly sure how it worked. So we'd gather their skin cells and then turn them into pluripotent stem cells, and then turn those cells into the type of brain cells that were dying from the disease called dopaminergic neurons. And we use that as a platform to study the disease, and then with the idea of hopefully, as Sandy and Shinya had talked about before, using that as a platform to discover new drugs. Over the last 5 years, I've been in on the evil capitalist side of the world, but I've also been taking, I call them soul supplements, by continuing to do a little bit of research at Stanford. And I published my most recent paper in July on using IPS and dopaminergic neurons and also transfecting them with an optogenetic channel. So this is a light-activated ion channel. So you can shine a specific wavelength of light on the cell, and it turns it on. So you can just blast it at a certain frequency, and the cell just turns on. And then you can and what we did was we took those cells from a human Parkinson's disease patient and transplanted them into the brain of a rat. And then we run a fiber optic cable through the back of the rat's head, and we can use that cable to turn on and off the human cells in the animal's brain, and then start studying of how these human neurons integrate into the host brain. And so that was the first really foray into the power that stem cells have in a therapeutic capacity. Now, this is very far from actually wanting to put any of these in humans. We're not like going to be transplanting these in and running fiber optic cables anytime soon. The cells don't integrate into the native network as beautifully as the human, or as the brain does when it develops. They're this bolus of cells, and they kind of shoot tangles. There are axons everywhere. But it gave you a sense of, like, wow, these are human cells that we're transplanting into a rat, and it actually does something. It integrates, and we can turn on these cells and watch how the rest of the animal's brain performs. This is something that's much more sophisticated in response than what we've ever done before. And so I want to put like a disclaimer on what I'm going to talk about. I'm not going to talk about all the challenges that face stem cell therapy, because there are many, and there's many challenges that we'll need to overcome, but we will overcome them. And so instead, it's the question of, why should we do this if it's going to be hard? Like, why do the hard thing? And what is the promise of stem cells that our existing techniques don't promise before? So you can think about, we already have many types of drugs. This first small molecules were really developed by Ian Fleming in the early 1920s, but it wasn't until the kind of progress in synthetic organic chemistry that allowed us to produce that penicillin at scale. And that took another 15 years. Likewise, we moved from small molecules to larger molecules, proteins, and we first started using proteins to treat people also in the early 20s. We would ground up cow pancreases and isolate insulin and deliver that to diabetic patients. But it wasn't until some of the researchers right next door here and at Stanford, Herb Boyer and Stanley Cohen's work on recombinant DNA, that we're able to tune proteins to exactly what we would want to treat human disease. Likewise, with cell therapy, we've actually kind of been doing cell therapy in some ways for a while. We've been doing transplants, bone marrow transplants, blood transfusions for decades, but we just haven't really had the tools or the knowledge that we could shift these cells in a way that we could tune them to treat a specific disease that we were interested in. So then the next response is like, well, what can we do with that? Why work on these stem cells if they're so much harder to work with than just a small molecule, some chemical that we bake up in the laboratory, or a protein that we just have a bacteria produce for us? And the reason is because cells have something that none of these other molecules do. Cells have logic embedded into them. They have an ability to respond to their environment, integrate that response, and come up with their own intervention on our behalf. So if you take the standard of care, for example, in diabetes right now, is Patient has to monitor their own glucose levels by taking a blood prick and then inject themselves with insulin. We're kind of relying on them for this feedback cycle. If you take the cell therapy approach to that, it would be we put a cell into the body or engineer endogenous cells, the cells that already reside in the patient's body, to monitor the blood glucose levels themselves and say, okay, the blood glucose levels get above 100 milligrams per deciliter, then now suddenly create insulin and secrete it out of the cell. And the cell just does that for us. That's what our cells in our pancreas already do for us right now. We could never design a drug that had that exact same effect with that amount of logic built in. That's even a simpler case. Imagine one that's even more extreme. As Sandy mentioned earlier, there's many different types of cells that are already in us. So we all brought adult stem cells here with us today, not in our pockets, but residing throughout our body. So we have hemopoietic stem cells that reside in the long bones of our body, and they give rise to our blood and immune system. But what happens when something goes wrong? Right now, we've developed a number of different drugs to target diseases of the blood and immune system, but they can have just one specific effect. We put the drug into the patient, and there's a giant bolus spike of that drug, and maybe every morning a patient takes that drug, and it just has that effect, and then it peters off over time, has an effect, peters off over time. It's a pretty crude way of actually trying to treat a disease. In a hemopoietic system, you can imagine either taking new hemopoietic stem cells, kind of fresh ones, or you could induce the endogenous ones and fix them somehow so that they respond to the problem themselves. And if your body's not making enough white blood cells, they detect that and then start making more white blood cells for you. If your body's not making enough red blood cells, they detect that and make it for you. It's a level of sophistication that we would never be able to achieve with small molecules and proteins on their own. And that's really, when you cut down to the basis of it, because they have logic built into the cell. And so for us computer nerds, that sounds awesome, right? That's basically the operating system for engineering human biology. So for the first time ever, we can start thinking of much more sophisticated interventions than we were able to do before, which means we can treat and cure diseases that we were never able to cure before. If you think about the difference between delivering a drug over time versus a bone marrow transplant, one is a sequential series of treatments, basically indefinitely, and the other is a one-time or a three-time injection of some cells that then do the work themselves for eternity after that. The other advantage of thinking about cells as a logic gates is we can start to think about things that biology doesn't even do yet. So not only can we cure diseases as they arise, but we can start thinking about prevention of disease before it arises. And we don't even have to get too exotic in our philosophizing about it, because we already know some circuits that we would like to target. There's a huge amount of variability between everyone in this room and everyone in the world. We respond to the development of diseases in different ways, and we develop diseases in different ways. Some of us are more prone to some diseases or less prone to the others, and we can find out what's unique and special in those gain of loss or functions in some people and deliver it in a way of engineering and programming someone else to have that similar effect. So that could either look like variants of PCSK9, so you can reduce cholesterol levels in people, so no longer would you ever have to take a statin. You just naturally have much lower cholesterol levels, and thus massively reduce the risk of cardiovascular disease. We could also engineer in the ability to detect cancer cells arising in the body much earlier before they turn into large tumors that are then difficult to treat. And all this sounds, you might be like, oh, that sounds totally crazy. What are you talking about? But in some ways, we're already doing some of these things. So like we said about hemopoietic stem cells, we already been doing hemopoietic stem cell transplants since the 1950s. There's lots of problems with them, graft versus host diseases, et cetera, but they're problems that we will be able to overcome, and it will massively expand the set of diseases that we're able to treat with that therapy. If you think about hemopoietic stem cells themselves, it's not a complicated process to even transplant. your hematopoietic stem cells that arise in your body, about 10% of them every day bud off from your bone marrow and migrate around and reconnect in your bone marrow somewhere else. So it's a very easy, relatively easy transplant. You just inject them in, they know where to go, they home to the right endogenous niche, and then they start replicating themselves. Just a handful of a small number of hematopoietic stem cells can reconstitute and repopulate your entire blood and bone marrow system on their own. On the cancer side, we're starting to do that in a way that Sandy and Dr. Yamanaka described as well, where we can take cells out of the body and kind of train them to look for cancer and put them back in. And then they go and hunt for those cancer cells and eradicate them. This work is being done by many labs. There's a number of companies now that are public companies that are valued in the billions, which gets capitalists like me very excited. And it's just the beginning of a new field on the cancer side. So even though it sounds kind of fantastical, we're going to like take these cells and engineer them up and have logic gates in them and operating system. This all sounds reasoning by analogy, and it must be completely crazy. We've been doing some forms of it for decades and other forms of it relatively recently with great success. And so in some ways, the way that we saw small molecules took more than a decade before we could develop synthetic organic chemistry to make it broadly applicable and engineered. And then recombinant DNA for engineering proteins took multiple decades from using the first human insulin to now the variety of different biological molecules we have treating human disease. It's really been in the last 10 years where we've had the techniques for engineering and tailoring cells that we've never had before. And Dr. Yamanaka and Dr. Shiristava, who are here in the front, are some of the pioneers of that field, both in reprogramming somatic cells, adult cells, to pluripotent cells, as well as adult cells to other types of adult cells or other types of adult stem cells that we might be interested in using to treat the disease. And so kind of the conclusion of that is there's going to be lots of stuff that's difficult about this process. It's going to be hard, but it's going to be worth it. So that means we should try to do it, and that's the exact reason that that we're excited to be working in the field and very actively looking at companies in this general field of stem cells attempting to cure disease. I should say that Dr. Yamanaka told a story about his dad, and so I was sitting there thinking, well, my dad's in the audience. I better tell a story about my dad too. And I think I have better hair than him, so I can't use the same joke. But my dad moved to San Francisco. I should also mention that my family's there, my brother and my mom, my cousin as well. And I definitely wouldn't be here if it wasn't for the both of them raising me. But I feel extremely lucky to be raised in this family. But the specific story here is when my dad was living in San Francisco in the 70s, Genentech was actually incubated basically out of his apartment. His roommate started Genentech while they were living together. And I was like, God, that's such a cool story. And I don't want to be telling your stories as my cool story. So I got to develop the next cell therapy company so I have my own cool story. I don't have to be regurgitating your story at podiums all the time. So a less sentimental one, but a true one nonetheless. So thank you.
All right, we're going to do two things before I open the floor to questions for Shinya and Blake from the audience. One, we're going to have a little moment of celebration. One of our own, Dr. Deepak Srivastava, has been elected and will undergo his induction ceremony into the National Academy of Medicine this weekend, making him the latest Gladstone scientist to achieve what is really a very fine distinction. I also thought it would be appropriate to ask Deepak to stand up and make a comment or a question to our speakers. Yeah, I would ask all of the questioners tonight to wait till you get the microphone. That way, we're videotaping this and we can be sure to immortalize your words.
Thank you, Sandy. So, one of the things, listening to both of you speak about the science and the business trends, it made me think about the technology revolution that we all know occurred here starting in the 90s in the Bay Area. And there was a bubble, maybe, in the tech industry in the late 90s, early 2000s that popped. But that didn't stop the long-term trend of technology fundamentally altering how we live our lives. And so I wanted to hear from both of you how you feel about the current situation where science has sort of been developing along, building a foundation for several decades now. Some people think that there's a biotech bubble, and we have many biotech folks in the audience here, because there's been so much interest in that area. And that may pop, but do you feel like that this is part of a long-term trend that now maybe will extend for decades, that it will fundamentally alter how we operate, or is this a different feeling? And I think many people in the audience might have opinions about that as well.
So I probably have the young, naive person's view of bubbles, where it might be a local maxima, but if the technology is sound, it's not a long-term maxima. So I find the only thing that's really true is the status quo doesn't stay the same, but as long as the trend line is up. And there's clearly many types of diseases that we are not close to treating, but we're going to treat them. And so it's just a question of the steps that we need to take to get there. And until we decide just to completely give up on scientific progress, there's almost a near infinite horizon of discoveries to be made. So I don't really, we don't concern ourselves too much with bubbles, especially on or price fluctuations in the public markets, especially on the venture side, because our timelines are much longer. It's definitely, we think about it as more concerning as our companies are preparing to go public, and we have to plan accordingly and be prudent in our business practice. But by no means are we slowing down on thinking about scientific progress because of what might be a short-term funding gap.
So as you know, we Japanese experienced a very tough time, the burst of economic bubble 20 years ago. But I don't think that's the case in science. I believe we will keep expanding. So 7 years ago, we had reprogramming, iPS cells. 2 years ago, we had CRISPR. 8 years ago, I couldn't expect iPS cells. 3 years ago, I couldn't expect CRISPR. So science is still a lot of space for expanding. We know probably only less than 10% of our life, of nature. So the remaining 90% is still in front of us. So I believe science will keep expanding, and there should be many opportunities for new cures and for new investment.
Okay. I'm going to ask just a couple of questions myself of our speakers, and then I'll open the floor to you. So one question I'd ask both of you is, what kind of gap exists between what we know now and what we need to know to use stem cell biology to really advance medicine? Is that gap relatively small, or is that gap still quite large? So we're going to need a lot more fundamental science for the really important applications of iPS technology to become real.
Yeah, well, I think this goes to the point you made earlier, but there's different forms of stem cell therapy, whether or not that's creating new types of stem cells and then transplanting that into people, or delivering something that switches endogenous stem cells to either turn on, or other cells that aren't stem cells to become stem cells and elicit their therapeutic function. And I think both cases we're actually, I think, much closer than even we were just 2 years ago. I think that's especially true in the case that I mentioned earlier in hemopoietic stem cells. And that's because unlike an organ like the liver, where you need dozens or maybe hundreds of different cell types all acting in concert, integrated into some large three-dimensional structure that's vascularized, innervated, and then we need to go plug it into somebody. Here we just need to know how to make this one cell. We just need to know how to make a hemopoietic stem cell, and then we already know how to transplant it. We just inject it into somebody, and then it just goes and does its thing. So it's actually quite a tractable problem. Like, can we make a hemopoietic stem cell? And there, in the last 2 years, just as Dr. Yamanaka had mentioned CRISPR, there's been other advances in our ability to produce that exact cell type. And this was something that we didn't just start thinking about in the last 2 years. It was an area that I was been interested in for 8 years now, and just kind of been biding my time waiting for an interesting project to come up. And we're actually, Google Ventures is funding a research lab out at Harvard that's working on this exact field. So we think it's like a slightly too early actually for a company. And so we started funding the lab with our dollars, just kind of granting it to the lab. And that started about a year ago, and it's already progressed even faster than we thought. So I think there's still a lot to do, but it's kind of, it's not unknowns. We actually know the exact experiments we need to do and the problems we need to overcome, and it's a really exciting time.
I agree with Blake. So there are many gaps, hurdles, but probably the most important step is to make cells of interest. We need to make brain cells or heart cells in order to make new cures. At this moment, we can make cells that are partially similar to our heart cells or brain cells, only partially. So we can make brain-like cells or heart-like cells. For some applications, those like cells are good enough. But for many other applications, we really need cells that are indistinguishable from our brain cells or heart cells. So there are a big gap between what we are now and what we should reach. So many scientists have been working on that gap, but so far, nobody has overcome that gap.
Very good. Okay, it's your turn. And if any of our previous exercises like this serve to inform what will happen tonight, I expect some exciting questions. So please.
Hi, thank you. I've been seeing a lot of these programs dealing with T-cell or gene manipulation, where they're taking cells, infecting them with the measles or AIDS virus, and then bringing them back into the host body and curing cancers, seeing some pretty miraculous results even with the human trials. Is that related to kind of what you guys are doing? And there was some talk of FDA fast-tracking this process to be available even by 2016 for some forms of cancer cure type thing.
Yeah, that's a little bit of the example I mentioned on the CAR T side. And so there's two public companies called Kite and Juno that are working in that field. And then there's a number of other young companies behind them that are already doing the 2.0 version of what they're doing, even though it's not an approved therapy yet. But the response rate from those types of trials that you're mentioning is really unprecedented. And it kind of goes, I think, it was surprising at first, but it almost makes sense when you think about it. Before, we were just delivering this molecule that was supposed to hone to specifically only cancer cells and only eliminate them, and somehow eliminate all of them and leave the host body relatively unchanged. That's asking a lot of a small little molecule, right? But cells are built to recognize different things. They have millions of different potential sensors that they could develop themselves to go, in this case, antibodies or recognizing the antigen of interest. And they can hone to that specific cell type, and they can be engineered to do so. And so I think that's, right now one of the most promising real world examples of this.
Yes, as I mentioned in my talk, we are very excited about T-cell-based cancer therapy. As you may know, we now have PD-1 inhibitor, which enhances the activity of T-cells. And that one drug, PD-1 inhibitor, is now causing a miracle for like 10 or 20% of end-stage cancer patients. They can survive. But for the remaining 80%, it doesn't work. We don't know why. But we are learning that T cells are very powerful in curing cancer patients. So we really want to combine our technology and T cell technology to find more cures for more cancer patients.
Sandy, you mentioned earlier CRISPR. I know you have a 1-hour speech, but could you do a 2-minute description of what CRISPR is, please?
Sure. Is my microphone on? Yeah. Oh, good. If you're not familiar with this, I think many of us feel the revolution that IPS technology created just a few years ago is now being followed by another revolution that had its origins right across the bay at Berkeley called CRISPR. I won't give my hour talk on this, but I'm happy to if invited, because we're using this technique every day at Gladstone, and it opens up some amazing new opportunities to accelerate the path to cures. It's a way to go into a cell and change its genes. The IPS technique that Shinya pioneered takes genes and packages them in a virus and then moves them into a cell. Those same genes are already in that cell, but they're turned off. They're not activated in the way. By putting them in from the outside, they're master regulators, and they then turn on all this other complicated program of genes. What CRISPR does is actually go into the genome and change the genes that are there, and it does so 10 to 100 times faster, cheaper, and more accurately than any technique that we've had before. So this is the next revolution. But there's a big word in science now that I really like, and we practice this all the time at Gladstone, that I call convergence. We have a convergence zone now. What convergence means is taking one revolution and matching it with the second one, and the combination of those two, and then the third one and the next one, really creates a powerful accelerant. So converging IPS technology with CRISPR-based technologies is really the hottest thing going.
Yeah, and to that point, we actually have a portfolio company that's working on CRISPR therapeutics. So kind of the early potential indications are, taking someone's blood cells or some cells that might have something small wrong in the DNA, and then using CRISPR just to fix that problem and then deliver it back to the patient. So already an area of active development on the commercial side.
Questions?
How close are we to actually taking what we know about stem cell therapy and macular degeneration and actually beginning to use it on patients, finding a vehicle to deliver the stem cells and get real clinical results?
Well, that's a very super question to me, because in Japan, my friend, my collaborator, Masao Takahashi, has already started clinical trial using iPS cells for patients suffering from age-related macular regeneration. So far, she has done only one patient. The patient has been very good. She is now preparing for second patients. The biggest hurdle right now is the cost. It costed almost $1 million just to make iPS cells from that patient and to evaluate that iPS cells from the patient. So it was way too expensive. So we are now trying to cut the cost in order to make it more widely available to as many patients as possible.
Perry?
So I was going to ask something somewhat similar, but for those of us who've been in the field for a while on the commercial side, I think we've set expectations that haven't quite yet been realized and maybe caused some frustration, both on the commercial side as well as among patients who are waiting for cures. You know, I totally agree with all of you on the engineered T-cell side and what's happening with immuno-oncology. Those are significant advances. But besides the patients, Dr. Yamanaka, in Japan and macular degeneration, when do you think we will see the use of either iPS cells or transdifferentiation or other cell types to truly bring cures to patients in regenerative medicine?
So, you know, many applications are very close to clinical trial, actually. For example, we can now make functional platelets, blood cells from iPS cells. And I believe, I hope as early as next year, we can start clinical trial. You know, it's very important in many aging society like Japan, because within 5 years in Japan, we won't have enough number of blood donors. So many thousands of patients would die because of the shortage of blood transfusion. So we do need good alternative to blood donor, and making blood cells from iPS cells just around the corner.
But I'll echo one thing you said, which is not to like overhype a field and overpromise, and you want to be accurate in your prognosis of what you're able to accomplish. And that's why I give the disclaimer at the front of my talk. I'm like, there's going to be lots of problems. It's going to be really hard. But like, why should we bother doing this really hard thing? And so science always takes longer than we think it's going to. And I think that is important to communicate to patients and not like overhype this word stem cells, because it sometimes results in consequences that none of us would expect would happen. As Sandy alluded to at the beginning of his talks, there's now like stem cell clinics popping up around the world. They're like, well, I can't get stem cells in the US, so I have to go elsewhere. And these universally are all very shady practices that patients always emerge from worse off than they go in. And definitely as a community, we have to be aware of that and communicate accordingly. So I couldn't agree more with that point.
Can I add a comment here? Oh, here, I have a microphone. There we- I had a wonderful experience a couple of weeks ago when I was asked to participate along with only 7 other scientists from all over the world in a think tank, asking the question, what are the biggest problems in biology that might be solved, at least to some degree, to affect medicine within a 10-year period? And this group of scientists who represented lots of different disciplines and lots of different institutions converged on the following question. How can we create the instruction manual for building a complex tissue like a heart or an area of the brain? And it was generally thought that though we can't do that now, the techniques are emerging that a concentrated effort to understand that blueprint. I like to use the metaphor of reading the machine language of the cell so that we can then go back and write the source code that would have the cell do what we want it to do, or get it started so that it then knows what to do itself. That this is a knowledge gap today for the really hard problems, but I and others view this as tractable in our lifetimes.
First of all, I want to say, as the mother of a 12-year-old girl with type 1 diabetes, she's had it since she was 2 years, 9 months old. I want to say that I am truly grateful for the work that Dr. Yamanaka has done in this area, and of course, Dr. Byers. Dr. Byers, you said something that I thought was very intriguing, and I'd like you to help me understand this a little bit more in the context of type 1 diabetes. And that is, you said that cells, the reprogrammed cells would reconstitute and repopulate existing disease cells. How does this work in the context of type 1 diabetes, where you have to regenerate or rejuvenate islet cells and at the same time reprogram cells to suppress the immune system?
Right, so I think there's two different potential approaches. One would be, and I should say at the beginning, like all the contributions to the field are definitely over on this side of the table, not this side of the table. I like a very small effect. So there's two different approaches. One would be there's still probably like a small reticent number of cells that are producing like a small basal level, or there's other cell types in the area that could potentially be converted into those beta islet cells that could produce insulin. This is similar to Deepak's work on reprogramming fibroblasts into cardiomyocytes. You can imagine something similar potentially of converting some other cell type in the area into an insulin-producing cell. Alternatively, you could make those cells ex vivo outside the body and transplant them in. Then you start to deal with a little bit of the element that you outlined of, okay, in both of these cases, how do we deal with the immune response to these systems? And that's kind of the interesting thing about these cells. Like we could potentially engineer in that immunogenicity to the cell itself, or that immunoprivilege to that cell. I'm not doing work in that area, so I don't know, specifically how close it is, but maybe Dr. Yamanaka knows more people working on islet cell production.
So we have a very strong collaboration with Takeda, which is the biggest Japanese pharmaceutical company. And there are at least 3 major programs working on type 1 diabetes. So I believe in all 3 approaches. We make insulin-producing cells from iPS cells or ES cells, and we encapsulate those insulin-producing cells in a device. And then we transplant that device, which contains cells, into patients, so that those insulin-producing cells are not attacked by patient's immune system. Otherwise, patients have somehow developed immune cells that attack insulin-producing cells. So we need to protect newly generated insulin-producing cells from the same immune system. So I think the current approach is to protect by a device. So I cannot give you, I don't want to give you any hype, but 3 major companies are working, competing with each other to make it happen. So please have some hope.
Yeah. Yeah, I can add too, we do have a program in this area at Gladstone that involves 2 of our scientists. Sheng Ding, who's a chemical biologist, has identified very effective ways to turn human fibroblasts into glucose sensing, insulin producing pancreatic beta cell equivalents. And when those cells are transplanted into diabetic mice, they cure the problem. That still leaves the problem of the immune attack in humans. And another of our scientists, Eric Verdin, has a program to modulate the function of T cells so that they would allow the beta cell to be privileged and not destroyed. So we have a program in that field too.
We've heard today about CAR T cells and PD-1 and ways to augment the immune system to attack cancer. My question is, do iPS cells have a role to play with either helping to understand what is a healthy immune system or maybe someday helping to broadly regenerate a compromised immune system?
So there have been many attempts to use iPS cells in understanding immune system and also in treating cancer cells by immune system. So, for example, some researchers have succeeded in making natural killer cells from iPS cells. Other scientists, including one of my colleagues, he has succeeded in making T cells, which specifically attacks a cancer antigen. So through that kind of researches, we're hoping to contribute to cancer treatment as well. So I hope I answered, at least to some extent, to your questions.
Let me add a point here too, if I may. Our bodies are taking steps towards cancer all the time. There was a kind of shocking paper that came out a few weeks ago where biopsies of the eyelid in human beings were sampled, and the genes of individual cells in that skin was determined. This was in patients who were having their eyelids fixed for cosmetic reasons, so it was easy access to the tissues. The alarming thing is most of those cells were not normal. They had already taken steps towards the development of skin cancers, either melanoma or basal cell carcinoma. And so we're stressing ourselves as we age towards cancer all over. Each cell has a defense mechanism, and it's possible that we could improve on those defenses ultimately. And then the immune system is surveying the landscape and destroying. When a cell pops out and says, okay, I'm here, I'm a cancer, I'm going to take over, initially the immune system can eradicate it. As cancers develop, the fast mutation rate causes them to become invisible to the T cells, and a lot of these immunotherapies are correcting that invisibility. But I think it's here today almost that we can, it is here today that we can combat cancers, but the big picture for me is, could we improve the surveillance system and kill them before they start?
I think to that point, you can imagine a system where, like, as we age, our immune system starts to degrade a little bit. You can see that in the profile of the types of cells your hematopoietic stem cell system is making. And as Dr. Yamanaka alluded to earlier, you kind of rejuvenate by reprogramming to some extent. So you could almost rejuvenate and recreate a new, younger immune system that might be better endogenously just at doing this profiling. But you could also engineer in new types of systems into it to make it even better at surveying for cancer. And that kind of philosophy you can extend, and you can get into a little bit of like fantasy about what could you really do with a complete new immune system, right? And now it's not just surveying for cancer, it's also reducing your risk for specific types of diseases, or even more fantastically, there's these experiments where they take a young mouse and they engraft the young mouse to an old mouse. They engraft their circulatory systems. And it reduces age-related markers in the old mouse. So it doesn't just prevent the old mouse from getting older, it actually reverses aging in some of the tissues in the old mouse. And these are, we're not sure whether or not it's just from the hematopoietic system. It could also be a variety of other factors. But it makes you start to think like there's actually this is potentially a platform where you can really reverse even other age-related markers. Now that's like way out there, but it's there, right?
I would like to know that, are there any kind of patients from whom you cannot make iPS cells? And if that is the case, how do the iPS cells from one patient would be accepted in the other?
So that's a very good point. Basically, we can make iPS cells from everybody. The efficiency may vary, but basically we can make iPS cells from everybody but many diseases are caused by autoimmune mechanism, like type 1 diabetes, Parkinson, some forms of Parkinson disease. So if we okay, it's not a super good example. So many diseases are caused by genetic mutation. Many diseases. So of course we can make iPS cells from those patients suffering from some kind of mutation. And if we can make brain cells from the patient's iPS cells, but those brain cells have the same mutation. So if we transplant that new brain cell, they would become sick again. So that means we cannot use patients' own iPS cells. There are two ways to overcome that problem. The one is to use CRISPR, genome editing technology, to correct that particular mutation. The second is to use iPS cells from other donors, healthy volunteers, who does not have the same mutation. Both are very promising approaches. We have been working on both, but more. As for the second approach, in many countries, including Japan and also the States, we have been trying to make iPS cell banks. So we are making a panel of iPS cell stocks in advance from healthy volunteers. We need to worry about immune rejection when using cells from other people. But some individual, probably there may be one in this room, they have a very specific type of blood type of cells, so that iPS cells from that particular individual won't cause a severe immune rejection in other patients. So we have been trying to find that kind of specific individuals and making iPS cells from those specific donors.
Well, first of all, thanks to you, Sandy, and Gladstone for hosting this symposium, particularly given this wonderfully interactive format rather than the usual lecture and Q&A. My question relates to shot selection. So given the technical hurdles that you mentioned, what specific diseases, could you elaborate a little bit more on what specific diseases or categories of diseases you consider to be most scientifically tractable, given where the field currently is?
So, as I mentioned, there are two major medical applications, cell therapy and drug discovery. In terms of cell therapy, good target diseases are like Parkinson's diseases, spinal cord injuries, heart failure, liver failure, diabetes. So they have something in common. Those are all systemic diseases, but they are caused by just one type of cells. In the case of Parkinson's disease, it's caused by a loss of function of dopaminergic neurons, very specific brain cells. In the case of heart failure, it's caused by a loss of cardiac myocytes. We have more than 200 types of cells in our body, but in those patients, the remaining 199 types of cells are intact, just one type of cells. So by using stem cells, we can remake those specific type of cells, and we can transplant that type of cells back into patients. So that's the case with cell therapy. In terms of drug discovery, I think essentially all diseases can be applicable because all we need is a new drug. In this case, iPS cells are used just as a tool to find a drug. So patients using those drugs won't recognize they're using iPS cells because iPS cells are used just as a tool to discover new drugs.
Yeah, on that point, we invested in a company that used Dr. Yamanaka's early patents from iPS cells, and the company was called Hyperion, and then a couple years ago was brought by Bristol Myers Squibb, and it was developing a specific set of drugs for later stage neurodegenerative diseases. I always forget whether or not it's announced what they're working on. And so in that way, it's already, those programs are being pushed into the clinic right now. And that's more on the discovery using iPS as a tool. On the cellular therapeutics side, which is maybe the more kind of out there, interesting side, I think the nearest term is going to be, I feel like I'm parroting it, but hemopoietic stem cells. Just because they are this, once we've made them, then we're done. We don't have to integrate them in some complicated way into the host, into the patient. And in some ways we already do it from bone marrow transplants to patients. So now we are just changing the source from another person to cells that we're engineering. There's a series of things we still need to do. We don't really know how to make hemopoietic stem cells that well from other pluripotent cells. Are they the exact same as the cell types that are already in people? Are there little subtle differences, and how significant are those differences in the outcome of once you've transplanted them into patients? And these are all the questions that researchers are addressing right now. But once we have those answers, it's not going to be a long road to the clinic.
Okay, I think we'll have one more question, and
Steve?
then we'll all go out and we can continue this over some wine. Steve?
So you've talked a lot about the limitations of science and the timing of the limitations of science, but at a time when healthcare costs are skyrocketing, today more than 20% of our GDP, we're reducing the spend on research. And the dialogue seems to be dramatically disconnected. We're not connecting that the reduction of healthcare costs is increased spending on research. And so the limitation seems to be reduced funding at the very time we need increased funding. And I just asked all three of you for your response to that.
Yeah, so I think not only have we seen NIH dollars in real terms go down over time, the budget's been flat, but in real terms it's shrinking. The amount of dollars going to basic research has shrunk even more dramatically, where I was just meeting with a few professors at Stanford last week, and I think the estimate was from maybe 30 to 40% of the NIH budget to like 4%. And instead, it's shifted more into translational research. So there's overall a crunch in funding, and then an extreme crunch at the kind of basic research side, where it seems the furthest from the clinic, but might actually have the biggest impact decades later. And I think it's going to be a difficult problem. I think the biggest issue is we don't have an objective function in research. So how do we actually measure the output that we're having and what the impact is? How do you measure that kind of return on investment for society? And we don't have one. So as a former scientist and now capitalist, but I still complain for scientists, when you go and you just complain hey, we need more research funding, people are just stop, I'm tired of hearing of it. So we need a better case, and I think that case comes from an objective function of measuring what we're inputting and what output we're getting. But maybe there is no objective function for research. Maybe it's impossible to know what impact a discovery is going to have before it has that impact. So it's a really tough question, even though I still think we should increase funding for research, even though I don't have any data to support it.
Yes, the cost of new medicine is extremely challenging, important. We now have many good new medicine, but they are just too expensive. Like PD-1 inhibitor, one injection costs, I believe, at least $10,000 US, just one injection. We now have a new tablet for type hepatitis C. My father was killed by hepatitis C. But now we have very nice medicine. But this small tablet, very small tablet, costs $300, I believe. And so everything is too expensive, and many nations will bankrupt. So how to lower those costs? We seriously need to consider. You know, we cannot blame pharmaceutical companies too much because they spend a lot of money on development. I understand at least 50% of a huge money being spent in pharmaceutical company is for clinical trial, especially phase 3 clinical trial, at least 50%, maybe 60 or 70%. So I would say the best way to lower the cost of medical or new medical or cures is to lower the cost of clinical trials. I can see some unnecessary clinical trials. In the case of PD-1 inhibitor, they need to do clinical trial on one type of cancer, like melanoma, then lung cancer, then liver cancer. I would say it's kind of waste of time and waste of money. And many patients are dying while waiting those clinical trials. So I think there are many ways to reduce the cost of clinical trial. And also, I hope by using iPS cells from thousands of individuals, we can replace many clinical trials with human patients, reduce the number of clinical trials by using iPS panels from many individuals. So that's another approach we are thinking.
Yeah, I'd like to leave you with a couple of comments based on some calculations I did recently using the best available data I could find. I calculated that a new therapy, likely a regenerative medicine therapy based on cellular reprogramming, that could lower hospital admissions from heart failure by 50% in this country. So it's not a perfect drug, but it's working in half the people, would save our healthcare system $150 billion a year by 2040 that we'll otherwise have to spend if we don't have such a therapy by then. A comparable number for Alzheimer's disease, again, the same logic, a therapy is developed that's 50% effective, will save us as taxpayers and insurance payers by 2040 $500 billion a year that we'll otherwise spend on the ineffective treatments we have now. I have to believe that investing a few $billion a year now in the research that would have those kind of savings is a good investment. The other thing I'd say is if we're saving $500 billion a year, why not let the companies that make these drugs earn their 10 to 20 billion a year? That's a bargain. And that's why I think the companies that are driving innovation in medicine shouldn't be penalized or criticized as long as they're behaving well otherwise for the fact that these drugs cost a lot. It costs a lot to innovate. Okay, thank you, Blake and Shinya, and thank you. Thank you. That was